Good, good afternoon, everyone. We'll start in 5 minutes, okay? So that people can join in. Yeah, I guess we can start. So what we're gonna do today is a little hands-on things, that how we can take structure from the PDB, right? And then we can visualize it and understand certain structural aspects of it. Okay, so we're gonna do that. So for that, firstly, let me exit the screen. Yeah. And okay, it's right here. Let me share the screen. I hope you guys can see it, right? So it's the September month. You remember last time there was another molecule, and right now it's this molecule. Let's see what is this new molecule is. Molecule of the month is carbon capture mechanisms. Biocarbonate. Okay, so we'll probably start with simpler structure. So I don't have any such molecule in mind, but I guess we can explore something on the go. Should we go with SARS-CoV-2, insulin, let's say any transporter? Let's say, okay, we have this. Okay, because I want to explore the membrane protein aspect of this. Whatever other structural features. Okay, we can go with most likely all the receptors will be transmembrane receptors, right? That is why I just if you don't know the PDB ID of the structure which you want to see, you can actually write the keywords and then try to filter out the structures. And there are certain filters here. For example, I want source organism should be, I want only from Homo sapiens. Obviously it will be eukaryote only. Experimentation, I'm not that stringent, but we can go with maybe, let's say, X-ray, electron, right? Then I don't want to put any more such filters. These are the very basic filters if you want to filter them out. Otherwise you can directly go into different structures also. Okay, so this is how you can basically search things. Now a structure which I want to let you guys see or explore on the go is so this is a membrane protein structures. As you can see, it is a pentameric format channel, right? The organism from where this protein is Vibrio cholera, and the expression system from which this structure has been determined is Escherichia coli. So the first thing first, to visualize this structure, you can visualize it here also, right? As we have seen in the previous classes, that this visualizer works really well for visualizing basic things and information associated with the respective protein. But we gonna do a little bit more clear imaging, and we'll see how we can explore it. Okay, so let's see. So for visualization, obviously I need a PDB. PDB is an extension file which is a 3D structure file for tools to read, tools like VMD, Chimera, these tools, right? So I'll just download the PDB format of this, right? It will be downloaded very soon because the size is not that big. Okay, then we're gonna go and visualize it. So we'll visualize it in a tool called VMD. So let's see. Here it is. Okay. I hope. Okay, I think there's some issue with the VMD thing. Let me redo it one more time. Okay. This load. I guess on full screen there is some issue, so I'm gonna keep the screen to the maximum size I can do without actually keeping it in full. Yeah, let me just quickly make the background white so that we can see it more clearly. Now we're gonna understand the structure. Okay, so first, to understand the structure, I'll just okay, I'll share my whole window with you guys. Yeah, I hope you guys can see my screen. So this is the raw default representation of the structure. I am just modifying certain aspects of the 3D structure so that we can visualize it better. Okay. Right. So you can see something is different here, correct? Now it is more clear. So here is the protein. So this is the maximum window size I can do. I hope it's visible to you all on the full screen. So this is basically a 3D structure of a protein, which clearly you can see from here is pentameric in nature. By pentamer, what do I mean? I mean there are monomer units, so multiple monomers come together to make one functional protein. These monomers can either be same, which will be called homopentamer because there are 5 subunits, and if they are different in nature, they will be called heteropentameric structure. Okay, so this is one way of representing a 3D structure. So you can see, you can color it accordingly. So this tool which I am using, it's VMD, I guess version 1.9 if I am correct. So this is VMD, and it is mostly used for visualizing trajectories from the simulation, but obviously you can visualize the structure as well. Okay, so as you can see, it's a membrane protein, correct? So these are the N terminals of the protein and the C terminals. So this is N terminal and this is C terminal. So both N terminal and C terminal are in the same site. So consider this as a cytoplasmic site, and this is the outside of the cell, or towards the extra ECM matrix side. Okay, so this is the cytoplasmic side, this is the ECM side. So both N and C terminal are at the N and C are towards the cytoplasm, correct? As it's a transporter, It's from Vibrio cholera. So, and if you remember, there was a description also, right? It's a formate transporter. So it's a transporter which transports formate. So from where it will transport, if we don't know that, and if we want to understand one such phenomena, so one way is to first go with the experimentations, like different experimentation, what is the rate, how it is transporting, what it is transporting, all those things. Another way can be that if you can simulate the natural behavior of this transporter, provide the substrate based upon your literature review, right, and then you can have a basic idea about how this transporter or channel might be working. So for that, the starting material will be this 3D structure of a protein. Later you can add membrane, you can add water molecules, you can add substrate, and then simulate the whole system to see how it is transporting, whether it is transporting or not. Okay, so this is one thing. We'll dig more into it. Just give me 1 minute so that I can load more molecules into it, and maybe a trajectory, so that you guys can understand what I mean when I say that you can see the function of the protein by simulations. So can you tell me in the meanwhile that what are the features of this protein which you are seeing in front of you, right? So there are certain features. It's a membrane protein at the end of the day, correct? So in the membrane protein, what are the characteristics? Let me load one trajectory if I'm able to. Yeah, so in the background the molecule or the simulation is loading. Maybe we'll be able to visualize it. If not, then we can visualize it later. There was some space issue and hence I was not able to transfer the files, but surely by next class I will do that. Okay, so as I was asking, what are the basic features which you can understand from visualizing this tool? So it can be easily seen that this protein is majorly what? This protein is majorly alpha helical in nature. Okay, by alpha helical I mean you literally cannot see any beta strands or beta sheet structure in this, correct? Indicating that the membrane protein which we are seeing is predominantly alpha helical in nature. It is helping us understand that transporters, if we say for example this must belong to a family of proteins. So a family of protein which is able to transport small ions such as formate is predominantly alpha helical in nature, and it is pentameric in nature also, right? So now if we want to see what are the different secondary structures which are present in the corresponding structure which we are visualizing. So this is the color according to the secondary structure. You can see that, as I said, there are majorly what? Yeah, majorly alpha helices, right? So all the pink shaded region is the alpha helical region, while all the green color region is the loop region. So if you remember, while studying about the secondary structures, we actually studied about a loop which connects one secondary structure to another secondary structure, or it is just a stretch of amino acids which doesn't have any particular structure. Neither it is a beta strand nor it is a helix, right? So it is just helping in aiding connecting different structures. Here if we see, you can see if I went to the representation mode and I tried to color it with respect to the secondary structure which is reported in this 3D structure file, you can see that there are majorly 3 different types of secondary structure, but the predominant is alpha helix only. Okay, so this predominant alpha helix structure, what it is indicating, that it is pretty ordered structure and it is in the membrane. So you know, we talked about beta strand, that they cannot exist alone as strands, they need to be in the sheet like structure. But if you remember, when we talking about the membrane, in the membrane we said that it is highly hydrophobic in nature, the lipid membrane, correct? So the lipid membrane is hydrophobic in nature, indicating that whatever proteins will be inserted in the membrane should be energetically favorable. So if we put proteins which are completely polar in nature, or say water loving, they won't be able to stay in the membrane, correct? If they are not in the if they are not stable in the membrane, they won't stay in the membrane, correct? So for that purpose, we need proteins which has the ability to stay in highly hydrophobic environment which is present in the membrane. So to answer this question, how nature has done things is, they have made alpha helix as a predominant secondary structure. I am not saying that there are no beta sheet structures protein available in the membrane, but predominantly it is helix, and even in helix it is alpha helix. So in alpha helix, the property that the first residue is connected with the hydrogen bond to the nth I plus fourth residue, it helps in arranging the residues in such a way that every alternative phase of the helix is amphipathic. So what do we mean by amphipathic? If you remember, when we were studying about the helical structure, we actually saw the helical wheel projection, right? So in helical wheel projection, one side was completely hydrophilic and the other side was completely hydrophobic, correct? So in hydrophilic and hydrophobic, what happens is, the hydrophobic part of the alpha helix faces the membrane, right, enabling the stability of the protein in the membrane, while the hydrophilic part or side of the helix faces the inside of a protein. Hence, it provides a secure path for transporting molecules which are polar in nature. Remember your basics, like why on the first hand we need proteins in the membrane, because membrane is not allowing large polar molecules to move across them, but we want them to move so that it can act as a fuel for the cell, or it is required for the basic biochemical pathways of the cell. So we basically require a constant exchange of resources across the membrane, but membrane is not allowing that. So how does the evolution take care of it? By assuring that there are certain dedicated proteins in the membrane which will be able to transport these large polar molecules. Now the basic chemistry remains same. A polar molecule which is able to transport or transfer from any site across the membrane should pass the membrane in such an environment which is suitable for it, and that environment will be polar and hydrophilic, correct? If that is not the case, then it can pass across the membrane, and then we won't need any protein in the first place, if we are talking about transporters and not the function of proteins as receptors and all that, correct? So for transportation purposes, majorly transportation purposes, these proteins provide a energetically favorable passage for these large polar molecules so that they can transfer across the membrane. For them, they need to have a polar surrounding polar environment or polar cavity through which these molecules can pass. So how it is done? That is why helix secondary structure is suitable for membrane transporters, because it is amphipathic in nature. One side which is predominated with hydrophobic residues faces the membrane side. For example, these residues which are here. Let me see if I can put in bonds. right? So these are the molecules. So these are the bonds. So you can see that these are the residues. So certain residues are facing towards the outside, while certain residues are facing towards the inside also. Let me see if I can refine it better. Correct. So you can see this. So these are the helices. Certain residues are facing outside, certain residues are facing inside. So all the residues which are facing outside, they are the ones which are predominantly hydrophobic in nature, because they have to face the membrane, hence they have to be in the similar environment as the membrane allows, correct? While the inside of these channels, these parts are predominantly hydrophilic in nature. There are exceptions to this basic concept, but the overall phenomena or the net effect of these residues will be like this, correct? That is why helices are more stable in the membrane. On the other hand, if we have beta sheets or beta strands, so what happens is they also have amphipathic nature, but as they are extended strands, extended sheets, they are not that suitable to be in the membrane. Hence, predominantly most of the transmembrane proteins, irrespective of whether they are transporters, receptors, have enzymatic activity and all those six functions of our membrane proteins, irrespective of that, majority of the proteins will be helical in nature. Now let me try if I can show some movement of protein. Let's hope for the best that we can do that. Okay. Excuse me. Excuse me. Yeah. Okay, so there is some issue with this that I'm not able to start the simulation. No worries. Excuse me. It's okay. We can see the simulation by the next time, because I have the files right here, but there is something wrong. So we'll load it later, most likely. Yeah, we'll load it later. So yeah, so what other features we can learn from this protein? So this is simply what I'm doing is I'm visualizing a 3D structure. As I told you, VMD is predominantly used for understanding and visualizing simulation trajectories. So how a simulation trajectory work is basically not going into too much detail or technicalities. What we do is what we cannot do in experimentations. Okay, so if we talk about classical simulations, so in classical simulations what happen is we have a system, and by system what I mean, we are here if we are considering about the membranous protein, say this transporter itself, and I want to understand and see what happens if I just put its NAS. For example, if this is a insulin receptor, or say any say it's GLUT2 or GLUT4, these are transporters of glucose at the end of the day, right? So its substrate is glucose. So what is a system? What I will do is I'll take this protein, the 3D structure of it, I'll build a system. The system will contain a membrane, right? The system will contain water molecules. The system will contain the substrate, for example glucose here, okay? Wait a minute, I'm trying to visualize if I can do that. Just give me 1 minute. Okay. So you can see all this, right? So now let me hide the protein. So this is one system, right? So a system for me with respect to the protein or the type of behavior which I want to understand. I have these systems in place. Yeah, I guess now it's a bit more clear and visible, right? So if this is a system, all these ball-like structures are the head groups of one such protein. Okay, head groups means the phosphate head groups. If you remember the structure of the lipids, it has polar head group and hydrophobic long chains, right? So these hydrophobic chains or the acyl chains, it's depicted in line model. You can see here all these, right? And all these golden circles, they are the polar head groups. And in between, if I can show the protein again, it is the protein. So this is one such system. Let's say if I can show that also, so that it will be a complete so these are the things. Okay, so this is one complete system. So I simulated this system with a substrate which I wanted to understand how this transporter is transporting, and throughout the trajectory you can actually see how beautifully these molecule transport from one side to the another, and you can study, which is not possible if you are doing or studying in experimentation. First of all, it will take a lot of time setting up experiments and coming up with a hypothesis. On the other hand, if you are simulating a system, making a system with respect to the protein and its local environment, we can actually understand such phenomena in comparatively less amount of time. But one drawback is that these will be, in the end, will be just predictions, right, with high accuracy, in no doubt, but still they will be considered as predictions only, correct? So this is one such basic system. I hope you guys are intrigued with this. Let me see if I can find something else, and then we can move on to our next session or next part of the topic in the next class. So if you have any questions with respect to the system, how to analyze, handle, all these are subjective questions first of all, right? Because setting up a system, setting up a simulation, it completely depends upon what questions you want to answer. Okay, so if I stop sharing this, I'll close this window. Yeah, let me give a quick search to YouTube whether we can have a little bit of demo with respect to the simulation, right? Okay, let's see, we'll try to see this. Okay, I hope my screen is visible to you guys. Yeah.
And on early models, indirect experiments and everything, we have gone through a revolution the last 20 years. There are so many membrane proteins for which we now have not just structures, but high resolution structures that are so great that I can see lipids. In parallel to that, we can do simulations on time and length scales where I can actually simulate membrane proteins and see how a lipid will bind to them. And then we can do this. This is an old study of ours actually, but here we have part of a voltage gated ion channel where the orange solid parts are examples of lipids we found in a new X-ray structure, while these licorice parts here are lipids from the simulation. And we started them far away and then they fused and found their binding site. So it appears both the simulation and the X-ray structure predict that you would have lipids very tightly bound to a certain part of the structure. If they were not tightly bound, we would not see them in the X-ray crystal because they have to be the same conformation in every copy of the crystal. We can test that even more in simulations, because in simulations I can check how fast the lipids are moving and diffusing around the membrane protein. Remember Singer-Nicholson? Proteins should diffuse in a sea of lipids. That's not quite true if you ask the simulations. The protein here is white. I don't care about the proteins, just the lipids. The lipids far out here in red, they are the ones that are diffusing freely as if they were in a lake. But as I'm getting closer to the protein here, we turn into yellow, green, blue, and eventually black territory. And that means that the lipids are hardly moving at all. In fact, we have at least some of these in these cavities here, the lipids are so tightly bound that they never move. They're literally part of the structure. And even around that, we typically have one, if not two layers of lipid that is some sort of coat around the protein. So it's technically true that the protein is diffusing in lipids, but it is diffusing together with one or two layers of lipids around it. The lipids are effectively part of the protein. Look at the size here. The dark part here is almost twice the size of the protein itself. We can look at that another way, by checking the average jump length of lipids when they diffuse and see how that varies. As we get further away from the protein, things move faster and faster and faster. But it's pretty much just in the outskirts of this box, right, that we've reached a region where they behave like bulk lipids, lipids that are in a pure lipid bilayer. Anywhere close to the protein, we're going to have a significant influenced property here. And we can look at this as probability of moving a certain length, and here too we see that we need to get almost a factor 2 away from the protein before we behave like bulk lipids. Simulations are cool. This might not be numerically exactly correct, but the properties definitely make sense, and it shows that we have a much larger surrounding around proteins than we would expect just from looking at their structures.
Right, so it was a very basic
So looking at
basic demo of simulation. There are more videos in such a direction. If we want to understand more, let's see. This is quite a small video. At the end of the day, we can see this one more time, like this particular video once again.
a actual membrane protein structure, there are a couple of things worth noticing here. The thickness here, as I told you, was roughly 30 angstroms. And that means that if you do your math, you can count, well, including the head groups, we're going to need roughly 20 amino acids in an alpha helix to get through here. We can't have hydrophilic amino acids because if they're hydrophilic, they're going to love to be in water, and that would be very bad to expose. So whenever we find stretches of roughly 20 hydrophobic amino acids that prefer to be in alpha helical shape in a genome, that's a very strong indication that that's a transmembrane part. And in fact, that has historically been one of the way how we found membrane proteins. We identify them from the genome sequence and just predict that they appear to be a transmembrane helix. But there are these loops. Do you see how the loops are stretching out? There would be water exposed there, and there would be water exposed there. So it's not entirely hydrophobic. The parts that are here are hydrophilic. As we started to determine more and more membrane protein structures, initially many of us thought that if globular proteins are hydrophobic on the inside but hydrophilic, water-soluble on the outside, if you then have membrane proteins that are hydrophobic on the outside, it would make sense that they're kind of opposite, right? They should be hydrophilic on the inside. That was a very good idea, but it just turned out to be wrong. Because when these insert, they need to insert one helix at a time. I'll get back to that. And it turns out that membrane proteins are really hydrophobic everywhere. Most of them, at least. There will be some exceptions, as you will see later. So membrane proteins are not really stabilized by the hydrophobic collapse that globular proteins would be. They need to be hydrophobic to insert in the membrane, but then it's going to be a matter of packing all these helices in an efficient way, which is mostly Lennard-Jones interactions, actually, rather than the hydrophobic effect. There will also be some special effects up here. You might have a large domain bound here, and then somehow we need to create this membrane protein and insert it in the membrane, which is far from trivial. I can't dig a hole in the membrane and shove in the entire protein, because these proteins, they will not even be stable outside the membrane. They wouldn't fold until I have inserted them in the membrane. The other parts to think a little bit about is what happens with the residues in here. In some cases, we can actually have slightly charged residues. Let me show you a movie that this was made by Anna Johansson, a student in the lab several years ago. So if we zoom in here, do you see here how the lipids are moving? Because now we have actual lipids that are somewhat chaotic. I think this is probably 10 nanoseconds or so. The waters are moving everywhere. The waters love to form hydrogen bonds with head groups. And in this particular case, I have a membrane protein that contains a lysine. And lysine is not something we would normally expect to see in a membrane protein, but we can actually put a charged side chain either here, because this is effectively up in the loop, or even slightly further down, because this side chain will be able to stretch out and interact with the water.
Right, so I hope this will give you a very basic idea about what information we can take from the simulations. So this was just a demo, and even today's class was just for you to get familiarized with the simulations, what exactly is a simulation, what is a system, how we can see things, how we can understand things, what information we can take from it. It all depends upon what is your problem statement, what is your question at the end of the day, right? So we can do multiple things. The simulations are really getting better and better as we are moving forward. You can use all these studies in actually and then helping in translating it into a product at the end of the day, right? So feel free to ask any questions with respect to the simulations, with respect to the computational biology, bioinformatics, and all that information. So hopefully we can have a proper simulation by the next class, and the information which we can take from there. Okay, so till then, thank you so much for your attention, and see you in the next class. Thank you.